Conventional cancer therapy. Currently available cancer therapies such as radiotherapy or chemotherapy are effective at treating cancer. However, they also affect cells other than cancer cells, causing unwanted side effects. Targeted cancer therapy. In modern medicine, doctors are attempting to use drugs that will more closely target the cancerous cells themselves by looking at specific genetic abnormalities of within each tumor. In order for a cell to grow and divide, the cell nucleus must receive a series of messages telling it to do so from both outside and inside of the cell. Unfortunately, in cancer cells, these signaling pathways are often constantly switched on, promoting unregulated growth. Role of tyrosine kinase. Tyrosine kinases are enzymes that play a key role in such signaling pathways by transmitting signals through a series of phosphorylation of proteins in a domino-like effect. MAPK pathway in healthy cell. One of the most commonly affected pathways is mitogen-activated protein kinase or MAPK pathway. As with all such pathways, the initial step requires binding of a ligand, in this case the epidermal growth factor EGF, to its inactive receptor EGFR. Once binding occurs, two receptors come together in a process known as dimerization. Once dimerized, the protein changes shape and allows the binding of growth factor receptor-bound protein 2, also known as GRB2, and son of sevenless, or SOS, forming GRB2 SOS complex. The next protein in the sequence is RAS. RAS is normally inactive and bound to GDP. By phosphorylating GDP to GTP, RAS becomes active. This phosphorylation and activation process continues in a domino-like fashion through several other intermediary signal transduction proteins, namely RAF, MEK, and ERK. Finally, at the end of the chain, a variety of proteins are phosphorylated that have specific roles within the nucleus to initiate and promote cell division. c-MYC is one such endpoint and functions as a nuclear transcription factor that will initiate DNA duplication. MAPK pathway in cancer cell. Different parts of this pathway are commonly altered in cancers, so that the signal is permanently on, ensuring constant instructions for cell division and growth to occur. Different cancer in different people will have mutations that cause changes at different points within the process. These are important steps as doctors now have therapies that can be used to stop the signals by targeting specific parts of the pathway. Therefore, it is becoming more and more important that doctors know which parts of the process are abnormal to give appropriate types of therapy. Some of the more common malignancies are already being treated in this way. For example, Herceptin treatment in breast cancer targets HER2, a variant of EGFR, and thereby reduces division of cancer cells.